the virus doesn't attack the brain first. A person can still believe they only have an ordinary viral illness, while the first stage of brain invasion is already underway. The fever feels familiar, the headache seems explainable, and the body aches still belong to the logic of a normal infection. There is no confusion yet, no seizure, no loss of memory, and no reason to think the brain is in danger. But in hematogenous neuroinvasion, viral particles are already circulating toward the endothelial walls that form the blood-brain barrier, the microscopic boundary that separates the chemistry of the blood from the fragile electrical environment neurons need to survive. The brain is not the virus's first target. The barrier is. Before a virus can destabilize memory, trigger seizures, or inflame the cortex, it must first confront the endothelial cells that line the cerebral microvessels. These cells are sealed together by tight junction proteins that act like molecular locks, preventing pathogens, toxins, and immune chaos from leaking into the neural tissue. As long as these junctions remain intact, the brain remains chemically isolated from the bloodstream. The person may still feel sick, but the neurons are still protected. The first real danger begins when systemic inflammation reaches the vessel wall. Viral particles in the blood stimulate immune signaling, releasing cytokines that activate the endothelial cells from the outside. What begins as a protective immune response in the bloodstream now becomes a threat to the brain's most important defensive structure. The endothelial lining becomes stressed, the tight junction proteins can begin losing stability, and the barrier starts losing the absolute selectivity that once kept the neural environment stable. This is the hidden beginning of viral neuroinvasion. The neurons may still be untouched, but the vessel wall is already changing. Microscopic separations begin forming between endothelial cells. Transport pathways become less selective, and inflammatory mediators start priming the barrier for permeability. The person still experiences only fever, headache, or fatigue, yet the first true neurological event has already begun at the level of the blood-brain barrier. This is what makes blood-brain barrier failure so dangerous under the pathology of fear framework. The first symptom is often not neurological because the first damage is architectural. The virus does not need to enter neurons immediately. It only needs the protective wall around them to lose integrity. Once the barrier begins failing, the bloodstream is no longer chemically separate from the central nervous system, and the brain's most protected environment starts becoming vulnerable to everything circulating in the blood. Around these stressed cerebral vessels, astrocyte end feet and pericytes begin sensing the inflammatory disturbance. These support cells normally help preserve barrier stability, regulate vascular tone, and maintain selective transport. But once the endothelial layer becomes activated by viral inflammation, the support system itself shifts into an alarm state. The barrier is no longer a passive wall. It becomes a biologically unstable battlefield where every immune signal can widen the opening. This is the most deceptive phase of viral brain invasion because the body still feels like it is fighting an ordinary infection. The patient may take medicine for the fever, rest for the fatigue, and assume the illness is still confined to the bloodstream. But the true danger is no longer the viral load in the blood. It is the moment a first microscopic failure appears in the barrier that was built to keep the brain separate from the rest of the body. By the time confusion, slowed thinking, memory disruption, or the first seizure appears, the real invasion may already be minutes or hours old. The most dangerous step was never the moment the virus touched the neuron. It was the earlier moment when the vessel wall first lost the ability to decide what was allowed to pass. In biological terms, the attack on the brain begins long before the brain itself shows the first symptom. What the blood-brain barrier really protects. The blood-brain barrier is not simply a wall. It is a living filtration system designed to preserve the exact chemical conditions neurons need for stable electrical signaling. Every thought, memory, movement, and emotion depends on microscopic control over ions, neurotransmitter precursors, glucose delivery, and fluid balance. Unlike most organs, the brain cannot tolerate sudden chemical shifts from the bloodstream. A small change in potassium, inflammatory proteins, or circulating toxins can distort neuronal firing and destabilize entire networks. The barrier exists to prevent the bloodstream from turning the brain's electrical precision into chaos. At the center of this protection are the endothelial cells lining the brain's microvessels. Unlike ordinary capillaries elsewhere in the body, these endothelial cells are fused together by tight junction proteins that eliminate the open gaps normally found between vascular cells. Molecules cannot simply drift between them. Every substance that enters the brain must pass through a highly selective transport system built into the endothelial membrane itself. This is what makes the cerebral vasculature fundamentally different from the rest of the body. The vessel wall is not just a pipe. It is an active gatekeeper. These tight junctions are the first true lock protecting the central nervous system. Proteins such as claudins, occludins, and junctional adhesion molecules create an almost seamless seal between neighboring endothelial cells. Under healthy conditions, this prevents viruses, plasma proteins, inflammatory cytokines, and most immune cells from crossing into neural tissue. The barrier is selective by design, allowing only carefully regulated nutrients, oxygen, and essential metabolites to pass, while excluding everything that could disrupt synaptic stability. Wrapped around these vessels are pericytes, support cells that help regulate blood flow, endothelial repair, and barrier integrity. They function as structural stabilizers, maintaining vessel tone and signaling to endothelial cells when the barrier is under stress. If the endothelial wall is the lock, pericytes are part of the reinforcement system that keeps the lock functional under inflammatory pressure. Their loss or dysfunction makes the barrier more vulnerable to permeability shifts, microvascular instability, and delayed leakage. Beyond the vessel wall, astrocyte end feet form another protective layer. These star-shaped glial cells extend specialized processes that wrap around the outer surface of cerebral microvessels, creating a neurovascular interface between blood supply and neuronal tissue. Astrocytes regulate water movement, ion buffering, neurotransmitter clearance, and vascular signaling. Most importantly, they help preserve the molecular instructions that keep endothelial tight junctions intact. The barrier is not maintained by endothelial cells alone. It is preserved by constant communication between the vessel wall and the surrounding glial architecture. This entire structure exists to protect the brain's electrical isolation. Neurons require tightly controlled extracellular sodium, potassium, calcium, and glutamate concentrations to fire correctly. If plasma proteins, immune mediators, or uncontrolled fluid shifts enter the interstitial space, the timing precision of neuronal signaling begins to fail. The earliest consequence of barrier dysfunction is often not direct viral injury, but loss of chemical stability inside the neural environment itself. This is why blood-brain barrier breakdown changes everything. The moment selectivity is lost, the bloodstream is no longer separate from the central nervous system. Plasma proteins can leak into white matter. Inflammatory molecules can activate microglia. Fluid can accumulate in extracellular spaces. And infected immune cells can use the opening as an entry route into the brain. The virus no longer needs to solve the problem of neuronal access. The barrier failure solves it first. The deeper fear inside this architecture is that the brain depends on protection most people never feel. No one senses their tight junctions working. No one notices astrocytes buffering ions or pericytes stabilizing vessel tone. The system remains invisible until it begins failing. By the time the first neurological symptom appears, the most important event may already be over. The barrier that once kept the bloodstream from touching the brain's electrical world has already begun to lose control. How viruses weaken the barrier. The blood-brain barrier rarely fails all at once. In most viral illnesses, the first damage begins as inflammation at the vessel wall. Viral particles circulating in the bloodstream trigger an immune response designed to contain the infection, releasing cytokines, interferons, and inflammatory mediators into the vascular system. What protects the body at first can become the first threat to the brain. The endothelial cells lining cerebral microvessels respond to this inflammatory chemistry by shifting from a tightly sealed barrier state into an activated stress state, and this is where permeability begins. The earliest structural damage occurs at the tight junction proteins. Claudins, occludins, and other junctional components depend on stable endothelial signaling to maintain their seal. Under sustained viral inflammation, cytokines such as TNF-alpha and interleukin signals begin altering the phosphorylation state of these proteins, weakening how tightly neighboring endothelial cells remain locked together. The junction does not instantly tear open. It first becomes less stable, less precise, and more vulnerable to microscopic separation under continued inflammatory pressure. Oxidative stress amplifies the damage. Activated endothelial cells and surrounding immune mediators begin generating reactive oxygen species that attack membrane lipids, junctional proteins, and mitochondrial systems inside the vessel wall itself. The endothelial layer starts losing metabolic stability, and once the cells become energy stressed, their ability to maintain tight junction organization weakens even further. This is the same principle seen in other neurological catastrophes. The barrier begins failing as a problem of cellular energy and structural maintenance before the visible leak appears. At the same time, matrix metalloproteinases begin entering the process. These enzymes are normally involved in tissue remodeling, but under inflammatory viral states, they can become pathologically overactive. Once activated, they begin degrading the extracellular matrix and basal lamina that support the microvascular wall. The vessel is no longer only losing its seal between cells. It is losing the structural scaffold that keeps the endothelial layer anchored and resilient under stress. This is the hidden architectural collapse that makes viral neuroinvasion possible. The barrier weakens from multiple directions at once. Tight junction proteins destabilize. Endothelial mitochondria lose efficiency. Oxidative molecules erode membrane integrity, and the extracellular support matrix begins degrading beneath the vessel wall. The virus may still be in the bloodstream, but the protective architecture separating it from the brain is already becoming biologically unstable. As the tight junctions loosen, permeability begins rising in microscopic gradients. At first, only small inflammatory mediators and plasma proteins leak through. This alone is enough to disturb the carefully controlled ionic and fluid environment surrounding neurons. Astrocytes and microglia begin sensing the abnormal extracellular chemistry, shifting into an activated state that releases even more inflammatory signaling back toward the vessel wall. The barrier is no longer failing in isolation. The surrounding brain tissue is now helping amplify the breakdown. This creates a self-reinforcing loop. Viral inflammation weakens the endothelial junctions. The leak activates glial alarm systems, and the resulting neuroinflammatory signals feed back into the microvasculature to widen permeability further. The virus does not need to force its way through a healthy barrier. It only needs to sustain enough vascular inflammation for the barrier to begin dismantling itself. The most deceptive part of this stage is that the person may still feel like they only have a systemic infection. Fever, headache, fatigue, muscle aches, and body weakness still belong to the expected logic of an ordinary viral illness. But inside the cerebral microvessels, the first neurological event is already unfolding at the molecular level. The blood-brain barrier is losing its ability to remain selective. By the time confusion, seizures, memory disruption, or encephalitic symptoms appear, the most important step may already be complete. The danger was never only the virus in the blood. It was the earlier moment when the vessel wall itself began dissolving its own protective architecture, converting the bloodstream from a separate compartment into a direct threat to the neural environment. The entry routes, How the virus crosses. Once the blood-brain barrier begins losing selectivity, the virus no longer faces a single route into the central nervous system. It gains multiple biological pathways, each exploiting a different weakness in the neurovascular architecture. The most direct route is the paracellular pathway, where loosened tight junctions create microscopic gaps between endothelial cells. These spaces may begin as tiny separations large enough for inflammatory proteins and fluid to leak through. But once permeability increases, viral particles can begin slipping between the cells that once formed an almost seamless seal. The barrier does not need to tear open visibly. A microscopic widening is enough. A second route is transcellular transport, where the virus moves directly through the endothelial cell rather than between adjacent cells. Under inflammatory stress, endothelial vesicle trafficking becomes less selective, and certain viruses can hijack receptor-mediated transport systems to be carried across the cell membrane, through the cytoplasm, and out the opposite side into the neural environment. In this route, the barrier remains structurally present, yet its transport machinery has already been converted into a delivery system for the pathogen. One of the most dangerous pathways is the Trojan horse route. Certain viruses, particularly those capable of leukocyte-associated spread, infect circulating immune cells such as monocytes, macrophages, or lymphocyte-like populations while still in the bloodstream. Once these infected cells interact with activated cerebral endothelium, they can cross the weakened barrier as part of a normal inflammatory response. The barrier is not breached by free viral particles alone. It is biologically tricked into allowing infected immune cells to carry the virus across as hidden cargo. The defense system becomes the transport system. Certain viruses take an even more direct approach by infecting the endothelial cells themselves. In this route, the vessel wall is no longer only the gateway. It becomes part of the infected tissue. Viral replication inside endothelial cells destabilizes tight junctions, disrupts membrane integrity, and increases local cell death, widening permeability from within the barrier architecture itself. What began as a protective vessel wall now becomes a biologically compromised entry point embedded directly into the brain's microcirculation. In some neuroinvasive infections, the choroid plexus offers another vulnerable route. This specialized vascular structure inside the ventricles produces cerebrospinal fluid and has a different barrier architecture than the cerebral capillary system. Certain viruses can exploit this interface, crossing into the CSF and then spreading through the ventricular and subarachnoid spaces. Once inside the fluid system, the infection no longer needs to cross deep white matter microvessels first. It gains broad distribution through the spaces that bathe the brain and spinal cord. Some viruses bypass the bloodstream entirely as a backup route through the olfactory system. Viral particles entering through the nasal epithelium can move along olfactory neurons and supporting pathways toward the olfactory bulb, allowing direct neuroinvasion without relying solely on vascular permeability. Even in episodes focused on blood-brain barrier failure, this route matters because once inflammation reaches the CNS, olfactory and vascular routes can reinforce each other, accelerating the spread of infection deeper into limbic and frontal networks. This is what makes viral neuroinvasion so dangerous under the pathology of fear framework. The barrier does not fail in only one way. Once the endothelial architecture becomes inflamed, the virus gains multiple simultaneous opportunities, slipping between cells, traveling through cells, hiding inside immune cells, infecting the vessel wall directly, entering through CSF interfaces, or reinforcing spread through neural routes. The brain is no longer protected by a single lock. It is surrounded by doors that are beginning to open at once. The most deceptive part is that the person may still only feel worsening fever, headache, fatigue, or mild confusion. Yet by this stage, the virus may already be beyond the bloodstream. It may already be moving through extracellular spaces, cerebrospinal fluid, or infected glial and endothelial networks. The first true neurological catastrophe is not the seizure or coma. It is the moment the pathogen successfully changes compartments from blood to brain, because once that boundary is crossed, the disease transforms from systemic infection into direct central nervous system invasion. What happens the moment it crosses the brain? The disease changes from bloodstream infection into direct neural threat. The first cells to sense this invasion are rarely neurons. They are the brain's surveillance systems, Microglia, Astrocytes, and paravascular immune sentinels positioned around the vessel wall and extracellular spaces. These cells are designed to detect abnormal proteins, viral particles, and shifts in the chemical environment. The instant the pathogen appears inside the protected neural compartment, the alarm state begins. Microglial activation is often among the earliest CNS immune responses. These resident immune cells rapidly shift from a surveillance shape into an inflammatory response state, extending processes toward the site of invasion and releasing cytokines, chemokines, and oxidative molecules into the surrounding tissue. What begins as a defensive attempt to contain the pathogen immediately changes the chemistry of the local neural environment. Synaptic stability becomes threatened not only by the virus itself, but by the inflammatory response that now surrounds it. Astrocytes respond next, and their reaction changes the architecture of the tissue. These cells normally regulate extracellular ions, neurotransmitter clearance, water balance, and support for the blood-brain barrier. Once they detect viral proteins and inflammatory mediators, they enter a reactive state. Ion buffering becomes less precise, glutamate clearance weakens, and fluid regulation shifts toward extracellular accumulation. The earliest neurological danger is often not viral destruction of neurons, but the collapse of the stable environment neurons required to fire correctly. This is where the first signs of encephalitis begin at the microscopic level. The brain tissue surrounding the entry point becomes chemically unstable. Cytokines rise, extracellular glutamate increases, and local edema starts forming as fluid and plasma proteins accumulate in spaces that were never designed to handle vascular leakage. The neurons may still be structurally intact, but the environment around them is already becoming hostile to stable electrical signaling. The earliest network effect is often slowing, not destruction. Frontal and temporal circuits begin losing timing precision as inflammatory molecules interfere with synaptic efficiency, and extracellular ion balance drifts away from normal thresholds. This is why the first noticeable symptom may be subtle confusion, slowed processing speed, delayed memory retrieval, or unusual fatigue rather than dramatic seizure or coma. The networks are failing before the neurons are visibly dying. At the same time, the virus may begin directly infecting neurons or glial cells depending on its tropism. Certain viruses preferentially target temporal lobe structures, limbic pathways, or deep relay centers. Once inside these cells, replication disrupts membrane stability, protein synthesis, and mitochondrial function, converting early inflammation into direct cellular injury. What started as barrier failure now becomes localized viral encephalitis. The surrounding white matter also begins suffering. Edema and inflammatory infiltration compress axonal pathways, slowing communication between cortical systems even when the neurons themselves remain temporarily viable. Signal transmission becomes less synchronized, and the person may begin experiencing difficulty concentrating, unusual emotional shifts, or subtle lapses in awareness that feel disconnected from the original viral illness. This is the most deceptive moment of the entire episode because the patient may still believe the fever is the main problem. The body still feels sick in a familiar way, and the earliest neurological symptoms may seem too vague to recognize as brain invasion. But the real catastrophe is no longer the viral load in the blood. It is the fact that the infection has entered the compartment where inflammation itself can destabilize memory, perception, and consciousness. By the time the first seizure, profound confusion, severe memory gap, or sudden personality change appears, the true invasion may already be well established inside the neural environment. The most dangerous step was never the visible symptom. It was the earlier moment when the virus first crossed the barrier, activated the brain's immune alarm system, and transformed a systemic illness into active encephalitic network failure. BBB failure becomes brain swelling. Once the blood-brain barrier loses selectivity, the danger is no longer limited to viral entry alone. The failed barrier begins converting the brain's tightly regulated extracellular space into a fluid-filled inflammatory compartment. Plasma proteins, water, cytokines, complement factors, and immune cells now move into tissue that was designed to remain chemically isolated from the bloodstream. The first large-scale consequence of this shift is vasogenic edema, the swelling that begins outside the neurons and gradually compresses the networks they depend on. This swelling starts in the white matter and extracellular spaces surrounding the inflamed vessels. Because the tight junctions no longer hold the vascular compartment closed, plasma leaks into the interstitial tissue and draws even more water with it through osmotic forces. What begins as microscopic leakage rapidly expands into visible tissue swelling. The white matter tracts that once carried fast synchronized signals between cortical systems now become physically separated by fluid, forcing conduction speed to slow even before direct viral destruction reaches them. The most dangerous aspect of vasogenic edema is pressure. Unlike other organs, the brain exists inside a rigid skull with almost no room for expansion. Every millimeter of added fluid increases intracranial pressure, compresses capillaries, and reduces perfusion to surrounding tissue. This creates a secondary injury pathway where inflammation and swelling begin causing ischemic stress in regions that were never directly infected by the virus. The barrier failure now amplifies damage far beyond the original point of entry. As intracranial pressure rises, the earliest network effects often appear as worsening confusion, slowed responses, reduced attention span, and sudden difficulty maintaining coherent thought. These symptoms are not always caused by viral destruction alone. They are often the first behavioral expression of compressed white matter tracts, reduced cortical perfusion, and distorted extracellular chemistry from the expanding edema. The ventricles and cerebrospinal fluid pathways now become critical. As swelling pushes against surrounding tissue, normal CSF circulation may slow, raising pressure further and reducing the brain's ability to buffer the expanding fluid load. The person may develop worsening headache, vomiting, light sensitivity, or altered consciousness, symptoms that can still be mistaken for severe infection rather than the early stages of intracranial pressure crisis. Seizures often emerge in this phase because the extracellular ionic environment is no longer stable. Potassium buffering weakens, glutamate clearance becomes less efficient, and compressed cortical networks begin firing with abnormal synchronization. The seizure is not simply a dramatic symptom. It is a direct sign that the barrier's chemical failure has now destabilized electrical timing across large-scale neuronal populations. The white matter suffers a second wave of injury here. Fluid accumulation physically widens extracellular spaces, compresses axons, and disrupts the timing precision required for fast frontal subcortical communication. The person may experience memory lapses, delayed language processing, fragmented attention, or sudden emotional disorganization as the pathways connecting these systems become mechanically and chemically unstable at the same time. The deeper fear in this stage is what happens if the swelling extends downward. As edema spreads, pressure can begin distorting the brainstem and the narrow anatomical spaces surrounding it. This threatens the reticular activating system, autonomic control centers, and respiratory regulation pathways. What began as a viral illness in the bloodstream can now progress into a pressure-driven neurological emergency where consciousness itself becomes unstable. This is why barrier failure is more dangerous than viral presence alone. The virus may enter through a microscopic opening, but the swelling that follows can destabilize entire hemispheric networks, reduce global perfusion, trigger seizures, and threaten life through raised intracranial pressure. By the time the person appears profoundly confused or loses consciousness, the most important event may already be complete. The failed barrier has converted localized invasion into whole brain mechanical stress. When the virus starts killing networks, the most devastating phase begins when viral invasion stops being a barrier problem and becomes a network problem. Once the infection spreads beyond the entry zone, it starts targeting the circuits that give thought its structure, memory its continuity, and consciousness its stability. The danger is no longer just inflammation around the vessels. It is the progressive collapse of the communication systems that allow different parts of the brain to work as one. In viruses with limbic or temporal tropism, one of the earliest and most feared targets is the limbic system, especially the temporal lobes. These regions are central to memory encoding, emotional tagging, fear processing, and the ability to recognize the continuity of personal experience. When viruses such as herpes simplex invade this territory, the first visible signs may be sudden memory gaps, emotional distortion, déjà vu-like episodes, or personality shifts that seem terrifyingly disconnected from the earlier fever. The patient may still be awake, yet the architecture of memory is already being disrupted in real time. As the infection spreads through temporal and frontal connections, executive networks begin fragmenting. Thought sequencing slows, attention drifts, and the ability to maintain coherent reasoning weakens. The person may answer questions incorrectly, lose track of conversations, or show abrupt disorganization in decision-making. This is not simple confusion from being sick. It is the failure of synchronized frontal subcortical communication as inflammation, edema, and direct viral injury begin dismantling the white matter roots that hold executive control together. The thalamus becomes another dangerous relay point. Because it coordinates sensory timing, arousal, and large-scale cortical communication, viral spread into thalamic circuits can destabilize awareness itself. Signals reaching the cortex become less synchronized, sensory processing loses precision, and the person may begin drifting between lucidity and profound confusion. Consciousness starts fragmenting because the relay architecture that stabilizes it is no longer functioning as a coherent system. Seizures become more likely as cortical and limbic networks lose inhibitory balance. Viral destruction, glutamate excess, extracellular potassium shifts, and inflammatory synaptic stress all lower the threshold for synchronized firing. A focal seizure in the temporal lobe may begin as a brief blank stare, a distorted smell, a memory flash, or an emotional wave before spreading into larger cortical involvement. What appears as sudden seizure activity is often the visible sign that the infection has already destabilized large-scale electrical timing across the network. At the cellular level, neurons now begin dying directly. Viral replication disrupts protein synthesis, damages mitochondria, alters membrane integrity, and activates apoptotic pathways. Surrounding glial inflammation amplifies oxidative stress and extracellular toxicity, turning the local environment into a biochemical trap where even uninfected neighboring neurons become vulnerable. The disease is no longer just invading the brain. It is actively erasing the circuits that once made coherent cognition possible. The white matter pathways connecting memory systems to frontal control regions begin failing next. Retrieval slows first, then continuity of thought begins fracturing. The person may lose track of where they are in a sentence, fail to connect recent events into a stable timeline, or show sudden inability to hold multiple ideas in working memory. This is the terrifying moment where the infection begins killing the network, not just the cells. As edema, inflammation, and direct neuronal loss spread, coma pathways come into view. The reticular activating system, thalamocortical loops, and diffuse cortical connectivity all depend on stable relay architecture. Once enough of these systems lose synchronization, the person may stop maintaining wakefulness, drift into profound encephalopathic states, or become completely unresponsive. The visible coma is not the beginning of the catastrophe. It is the point where enough of the network has already been destroyed that consciousness itself can no longer be generated. The deeper fear in this stage is that the fever was never the real disease. The true danger was always the progressive conversion of a vascular barrier failure into network disintegration. By the time the person develops seizures, memory collapse, personality changes, or coma, the virus may already be moving through the very circuits that define identity, continuity, and awareness. What is being lost is no longer only tissue. It is the architecture of the mind itself. The point of no return. The true point of no return begins when blood-brain barrier failure, viral replication, edema, and neuroinflammation stop behaving as separate events and merge into a self-amplifying collapse loop. At this stage, the barrier is no longer capable of restoring selectivity. The extracellular environment is no longer chemically stable, and the infected tissue can no longer preserve the network architecture required for recovery. What was once a reversible inflammatory invasion now becomes irreversible structural loss. The first irreversible threshold is diffuse edema. Once vasogenic swelling spreads beyond the initial lesion and begins compressing wide regions of white matter, the problem is no longer only viral damage. Rising intracranial pressure reduces capillary perfusion, distorts extracellular ion balance, and starves surrounding neurons of oxygen and glucose. Regions that were never directly infected now begin suffering secondary ischemic stress. The brain starts damaging itself through pressure alone. At the same time, the barrier collapse loop continues widening. Persistent cytokine release, oxidative stress, matrix degradation, and endothelial injury keep the tight junctions from resealing. Plasma proteins continue leaking into tissue, glial activation remains elevated, and every inflammatory signal further destabilizes the vessel wall. The barrier is no longer a failed defense. It has become an active engine, driving continued neural injury. Neuronal death now accelerates from multiple directions at once. Direct viral replication destroys infected cells. Glutamate accumulation lowers excitotoxic thresholds. Oxidative stress damages membranes and DNA, and ischemic compression from rising pressure begins starving adjacent networks. This convergence is what makes late viral encephalitis so devastating. The person is no longer threatened by one mechanism. They are trapped inside several catastrophic pathways that are now reinforcing each other. The most dangerous shift in this stage is the loss of whole network rescue potential. Earlier in the disease, localized inflammation or focal edema may still spare enough white matter and cortical relay integrity for partial recovery. Once diffuse swelling, widespread barrier leak, and multi-regional neuronal necrosis take hold, the surviving pathways are no longer sufficient to restore coherent network timing. Even if the virus is stopped, the communication architecture may already be too structurally damaged for full restoration of prior cognition. This is the phase where intracranial pressure crisis becomes existential. As the swollen hemispheres continue expanding inside the rigid skull, the brain begins shifting across fixed anatomical boundaries. Midline structures distort, ventricular pathways compress, and the downward force on the brainstem increases. This is the beginning of herniation risk, the final mechanical failure where tissue displacement threatens the autonomic systems required for consciousness, breathing, and cardiovascular stability. The visible clinical signs in this phase are often profound: fixed gaze changes, severe seizures, sudden loss of consciousness, posturing, or rapidly worsening coma. But these are late manifestations. The real catastrophe occurred earlier, when barrier collapse became self-sustaining and swelling converted localized. Infection into global mechanical and metabolic failure. The deeper fear is that this threshold often feels sudden from the outside. A patient may move from fever and confusion into seizures and coma over hours, creating the illusion of abrupt collapse. Biologically, the point of no return was crossed earlier. At the moment, The barrier could no longer restore selectivity, and the rising intracranial pressure began converting salvageable networks into compressed necrotic tissue. This is the final truth of viral brain invasion. The virus does not need to destroy every neuron. It only needs to push the blood-brain barrier, inflammatory cascade, and intracranial pressure past the threshold where the brain can no longer protect its own architecture. From that point forward, the disease is no longer progressing toward damage. The damage is already becoming permanent. The barrier failed before the symptoms. The most dangerous moment in viral brain invasion is rarely the first seizure, the sudden confusion, or the collapse into coma. It is the earlier moment when the blood-brain barrier first lost the ability to decide what was allowed to pass. Long before memory fractures, before the temporal lobes begin failing, and before intracranial pressure rises, the true catastrophe begins at the vessel wall. The brain was endangered the moment the bloodstream stopped being a separate world. What begins as fever, headache, body aches, and fatigue can feel deceptively ordinary because the earliest neurological event is architectural, not behavioral. The virus does not need to attack neurons first. It only needs the endothelial tight junctions to weaken, the paravascular support systems to destabilize, and the extracellular chemistry to lose isolation. By the time the first unmistakable neurological symptom appears, the real invasion may already be well established inside the protected neural compartment. This is why blood-brain barrier breakdown is the true hidden danger behind viral encephalitis, post-viral neuroinflammation, and many of the brain complications people search for when they ask how viruses enter the brain. The fever was never the main threat. The real danger was the moment the vessel wall itself began dissolving the boundary that keeps neural timing, ionic stability, and consciousness separate from the inflammatory chaos of the bloodstream. Once the barrier fails, every downstream catastrophe becomes possible. Viral particles gain access to neural tissue. Plasma proteins flood extracellular spaces. Edema compresses white matter. Seizures emerge from ionic instability, and rising pressure threatens the brainstem itself. The visible neurological collapse is often only the delayed public version of a microscopic failure that began much earlier at the endothelial seal. The deeper fear is that the first visible symptom may arrive only after the most important protective system is already gone. By the time the patient becomes forgetful, hallucinates, seizes, or loses consciousness, the blood-brain barrier may already have transformed from a living shield into an open route for infection, inflammation, and mechanical pressure. What is being lost is not only neurons. It is the protected environment that made coherent neural function possible in the first place. The final truth of this episode is that the virus does not need direct access to the mind at the beginning. It only needs access to the wall that protects it. Once the barrier loses control, the brain becomes vulnerable to infection, swelling, disconnection, and the collapse of the networks that define memory, thought, and awareness. This is pathology of fear, and this fear is real.
Pathology of Fear is a medical documentary series presenting clinically documented neurologic and pathological mechanisms for educational analysis.